Hi there, AP Bio. I'm going to walk you through another FRQ from the PowerPoint in the green folder. So right now I'm working off of practice exam number 1, which is on the AP Classroom if you want it, and I'm going through question number 1. I'm here in the office. It's kind of late, but I'm kind of productive at the moment, so Jackson's going to help too. I thought I'd show you where our kitty sits on our desk here. Hey. Oh, okay. Well, he's a little grumpy because it's late for him too. He's usually asleep. So, so this is the question, FRQ number 1. It's got an A, B, C, D prompt following it, and it's worth 9 possible points from this test. And this is one of the styles that you would be expecting to see, where this one is covering chemistry of life and gene expression regulation. I would say a little bit of enzymes in there too, which can go in chemistry of life or energetics, but it's there. Okay, yeah. So we're talking about RNA polymerase. RNA polymerase, remember that that's the enzyme that converts the message of DNA into RNA. So it's reading the DNA code and building and assembling the RNA nucleotides together to make our mRNA message. So they first did normal RNA polymerase here in figure 1, and it has a maximum elongation rate of about 12 nucleotides per second, or 12 bases a second. And then the experimental strain, this is an enzyme, right? RNA polymerase enzyme. Well, they changed one amino acid in that enzyme, and it's a whole lot slower in this experimental strain. So these yeast that have an RNA polymerase still, but it's not working as well, probably down here around 2 bases a second. And you'll notice that there are error bars on these graphs, look a little different than the last one. And here at the end of the prompt, they say error bars represent 2 standard errors of the mean. SE is standard errors, and X with the bar over it is mean. Yes, I know it looks like sex. We can all giggle. But that's what they're telling you. So for the AP Bio test, you don't actually have to calculate your standard deviation or standard error of the mean. But when you see it in a prompt like this, you have to know what that tells you. What does that mean? So because the 2 error bars of these do not overlap, there is a statistically significant difference in the average. And you can see that in the graph here too. In figure 2, they add this compound, amanitin. God, they're so hard to pronounce. Don't worry about it. You're reading it to yourself, right? So amanitin binds, like amanatee is what I want to say, but no. Amanitin binds to RNA polymerase's active site. So as an enzyme, if you block that active site, that enzyme's not going to be as effective. So you can see the wild strain's effectiveness goes way, 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 way down. I wouldn't even say that's 1. I'd say it's like half a base a second. And the experimental is slightly reduced, okay? So let's go forward, look at the questions. You've got these. So go ahead, and if you haven't done it, try this. Pause the video or pause the PowerPoint, time yourself for 25 minutes and see how you do. And then that's what this slide says. Oh, excuse me, maybe I need to go to bed. Underlines are no longer in red. That's taking too long. This slide has these links of these videos, so that's my placeholder there. And here we go. So in A, describe 3 structural components of an RNA nucleotide monomer. We've been doing this all year. You should know this in your sleep by now, but you don't have to be as specific as you may think. You've got 5 carbon sugar, phosphate group, and a nitrogen base, nitrogenous base. I think base wouldn't be enough, so I'm actually going to underline that because that's specific. You don't have to say ribose, like if you forget if it's ribose or deoxyribose. And you could, oh my gosh, excuse me, you could mention the 4 bases there. So that'll get you the 1 point. Second part of A here, it's a 2-pointer. Explain the role of RNA polymerase during transcription. So these are their 2 statements. Recall that this is the kind of stuff that we only learned about in our distance learning since school closed. So you should have this transcription graphic organizer, I think that's what I called it, filled out. This was from like early March. So if you don't have that, it's in the PowerPoints. You can go to the calendar and click on it yourself too. But they're kind of specific here. You don't have to write a novel. Notice these are one statement sentences that get to the point and answer, explain the role of RNA polymerase. You don't have to say like, oh, an A will be binding to a U, a C will be binding to a G, it'll be going in the 5' to 3' direction, it needed to attach with, like, no, none of that's there. Great that we learned about it, but it's a one-pointer, folks, so don't go too deep on that. Any of these would be fine, okay? Notice they do have things like mRNA growing, they're talking about covalent bonds, so it is like, what is its role? Like, it's not just like what does it do, but like what is happening in transcription. So I think I might underline this too. So they're looking for you to like make that connection. It's more than just like identify, right? Explain is to kind of like put the content into there that shows you know that stuff. Okay, so that is A. B, identify the dependent variable in the experiments. Well, look what they did. What did they measure, right? Dependent variable depended upon what you did. So it's the same in both of them. That one's pretty quick. It is the maximum elongation rate. What are they measuring? Identify a control group missing from the second experiment and justify the need for this control group. So looking back at the second experiment, they added amytin. Well, they added it to both of them. How do you know it's the amytin that did that? How do we not know those wild strain are like dead for some other reason, right? Experimental didn't really change much. So what's missing is another bar of like with amytin, without amytin, with amytin, without amytin, because this is a whole nother experiment. This is not connected to these, right? They're two things that happen, but it's not happening in the same experimental setup, okay? So that's what they're looking for in B, the second part here, appropriate control group, either one of the strains from the first experiment, but without amytin. So you could say, oh, the wild type strain without amytin compared against the wild strain with amytin added. You need to have that control there. That would kind of be your negative control if you're thinking about that. Like if I don't add this, I should not see a change, okay? Or they said, but there may be other appropriate controls. I honestly can't think of any. I said something like no amytin at first, and then after 5 minutes add it and measure again. I don't know if that would be possible. So, and then justify the need for this control group. So think about why we use control groups. Like if you have a group that has no, enzyme added at all, you're testing that, like, does the enzyme do a job? Yes, it does. Okay, change the amount of the enzyme from this and this and this, double it, or add the enzyme and an inhibitor, and so you're doing all these like scientific experiments, but like you should have something as your baseline, right? So that's kind of this part B's in these question number ones are all about like the scientific method, like designing the experiment. You're looking at the details of the experiment and why they did it the way they did. Here they're missing something, and you're kind of justifying why it is that you need that thing. So let's see if I can do this one in all 15 minutes. This one was a quick one. So part C, describe the effect of amytin on the maximum elongation rate of wild and modified RNA polymerases. So you go back and you look at the graph, and it's the effect of amytin. So you got to look over at this one here. Well, they went down, right? So this was apparently our normal one, and these are way less. So amytin decreases the maximum elongation rate for wild strain and does not affect the rate of the experimental strain. For that, you would have to look at the error bars here and here, right? Actually, I'm going to highlight that because that's something I didn't notice. I'm trying live happenings. It does not. Right, so this is the same, and this does dramatically change, decreases. Okay, I'm happy with that. My camera back over us. All right, and so you're not actually having to talk about any numbers. You don't have to get into the details of the standard error of the mean, but knowing what you know about those, you're describing an effect, right? So an overall trend. So you could say there is no change because if you look at the error bar here and this error bar, they're in the same range, but this is a dramatic drop for the wild type strain, so it's decreasing them. My tip for you during your AP Bio test, while you are actively testing on a screen, have some sort of clear ruler. If you've got it, a regular ruler will be fine, but clear, because you can put it like this orange box I put on there. That's because I couldn't find a stupid transparent ruler on the computer, but I almost misread this experimental strain as 3 and not 2, which would have given me the wrong answer for the second part here. So you want to be really careful. Reading this as 12 on the line is not a problem at all, but reading this one and reading these that are farther over, using that straight edge and carrying your eye over to the axes will help you be a little bit more accurate. So that's a little tip. And then determine the ratio of the average maximum elongation of modified compared to wild type. So if we're doing a ratio, we're doing modified to wild, okay? So the answer is 1 to 6 or 1 divided by 6 or typed out 1 to 6. And so we're looking at this pink part here. We have an average of 12, and then down here, like I said, an average of 2 if I carry my ruler. And then look, it's about halfway between 0 and 4. So don't go 12 to 2. That's an answer of 6. That would not get you the point here, right? You have to look at the way that they stated the ratio, and it was modified to normal, okay? So yeah, clear ruler, watch your ratio, things there. And let's see, D, state the null hypothesis for the experiment and provide reasoning to, okay, so state the null hypothesis. You can't just say the thing they did will have no effect. Use the language of the experiment. Go into what that experiment was talking about. So they have one like the modified RNA polymerase, which is their original experiment, right? What are they changing that they did? They not only, oh, and it's for figure 1, right? So they're not talking about amitin not having an effect. Move over now. In this case, they're talking about the modification of normal strain to experimental strain. So having a modified amino acid should have no effect in elongation rate, which is probably what this first one is. Difference between the two elongation rates is due to chance. That's another really safe one to go for on a null hypothesis. I also think, oh no, okay, modifying the amino acid with the RNA polymerase of the experimental strain will have no effect on transcription rate. I think that would also suffice for this question. For the next, I have the word effect, and up here affect, affected. This is just a little help. To effect something is a verb, yes. The effect is the noun. So effect starts with E, and there's an E in verb. Try that. Common mistake, but they wouldn't mark you wrong for that. Just thought it was a good point. And then provide reasoning to justify the claim that the change in the amino acid sequence in the modified RNA polymerase affected the shape. So first here in D, kind of connects to experimental design again. You are like, okay, they did this experiment. What would a null hypothesis be for this? All right. Statistically, we do see that there is a change, right? These two error bars, because of this one amino acid change, are very different. So provide reasoning why. And this is really similar to the first question we did here. The change in the active site would explain a decrease in elongation rate. Oh wait, is this modify affected the shape? Oh, I misread this one, but it's still working. You're kind of again connecting about enzymes and their active sites and their ability to do their job. But I'm guessing, I'm not seeing if the amino acids provide the shape of the active site. That's kind of, they're kind of connecting figure 1 and 2. So not only the problem of changing an active site and making it work not as good, but also the amitin can be binding and blocking. So this is some enzyme review stuff in here. I got about 5 seconds. I caught it in 15, so that's